How do we know that DNA stores the genetic information needed for protein synthesis? This was made possible through the study of bacteriophages. Bacteriophages are viruses that infect bacteria. Bacteriophages are simple units composed of structural proteins and DNA, and rely on the cellular machinery of infected bacteria for their duplication. They attach to the bacterial surface and inject their genome into a host cell. Once inside, the bacteriophage genome directs the production of many new virus particles within the bacterium. These newly produced bacteriophages are then released when the bacterial host cells undergo lysis. Bacteriophages are simple units that are composed of proteins and DNA. They therefore rely on the cellular machinery of infected host bacteria to multiply. What led to this important discovery? Two initial observations were made about bacteriophages. The first was that bacteriophages attach to the outside of bacterial cells and do not enter the bacteria. This was shown with electron microscopy in 1940. The second observation is the existence of a transition phase between the bacteriophage and the production of new bacteriophages in the cell. This remained a mystery for years until it was solved by scientists Hershey and Chase in 1952. They used an experimental approach that was relatively new to science at the time, where both DNA and proteins are metabolically labeled. This approach allowed them to observe where the labeled biomolecules were distributed during bacteriophage infection. The first step in this process involved growing bacteria, specifically growing E. coli, in the presence of radioactive sulfur, known as sulfur-35, or phosphorus, known as phosphorus-32. The radioactive sulfur gets incorporated into proteins, and the radioactive phosphorus gets incorporated into DNA. The bacteria are then infected with unlabeled T4 bacteriophages. This infection led to the lysis of the bacterial cells, which released newly produced bacteriophages with labeled DNA and proteins. For simplicity, we can summarize the experiments with bacteriophages that have both phosphorus-32 labeled DNA and sulfur-35 labeled proteins. However, in Hershey and Chase's actual experiments, DNA and proteins were labeled separately. Following this, the next step was to infect unlabeled bacteria with the radio-labeled bacteriophages and allow attachment to take place for 10 minutes. The solution of infected bacteria was placed in a blender, and the sheer force of blending detached the bacteriophages from the surface of bacteria. Then the blended solution was subjected to centrifugation. This resulted in a supernatant that contained empty bacteriophage particles containing sulfur-35 labeled protein but without DNA, and a pellet that contained heavier components like the bacteria with the phosphorus-32 labeled bacteriophage DNA. In the follow-up experiment, the bacterial pellet labeled with phosphorus-32 was washed to remove any externally associated radio-labeled molecules. The washed bacteria were then suspended in a growth medium, and the accumulation of new bacteriophages inside the bacteria was observed over time. The bacteria underwent lysis, which released bacteriophages that were capable of infecting other bacteria. These pivotal experiments demonstrated that radio-labeled bacteriophage proteins did not enter infected bacteria. Only the radio-labeled bacteriophage DNA entered bacteria and contained all the necessary information to produce new bacteriophage particles. This experimental evidence finally led to the acceptance within the scientific community that DNA stores the genetic information needed for protein synthesis. Although this experiment may not be considered as more conclusive than the Avery, McLeod, and McCarty experiment, it significantly contributed to convincing the scientific community of the role of DNA as the genetic material.